Chapter 26 is all about the eye, and those two guys right there are nocturnal primates. The one on the left is a tarsier, and the one on the right is an eye. And what I want you guys, why they're there, is because I want you guys to keep in mind kind of the big picture. So we really emphasize structure, location, and function, really basic, fundamental things. But don't lose sight that those basic things can have a large impact on a way a species exists or behaves, or can even define what a species is. And we're going to talk about a lot of specific structures, and we're going to show you some different images of different species in this chapter. And just keep in mind how something as basic as pupil size or pupil shape can have a large impact on how an animal behaves. And you should really be keeping that in mind throughout the entire semester, like why and how something so basic can have such a large cascading effect on a creature. So the way today's going to go, we're really going to focus on initially some of those external features, external muscles, some of the superficial structures within the eye. Then we're going to work our way to some of the optical components, and then end on the retina and how we actually see and how we perceive light. So when you're drawing these things, make sure that you label everything appropriately. The eye seems to be one of the drawings that people mess up a lot, just because there are a couple of things to label and spell appropriately, and there's not one image out there that has all the things that you need to draw on it. Just make sure you take your time and go through a checklist and make sure you draw everything and label everything appropriately and accurately. So the first external structure we're going to talk about is the lacrimal gland. And the lacrimal gland is all about tears. If you remember the lacrimal bone, it has that structure called the lacrimal duct on it. Well, lacrimal means tear, and the lacrimal duct is going to be the draining point for those tears into the nasal cavity. You can see the superior lacrimal canal and the inferior lacrimal canal, and those two things are where those tears are draining into your nose. And so you can imagine that if you watch a sad movie or you're just crying, you're just bawling, right? Your nose starts to run, and then it's just a hot mess with all that mucus and tears pouring out. And what's going on is those tears are being produced from the lacrimal gland. They're draining into your nasal cavity, they're mixing with the mucus there, and then that mucus tear mix is coming out. It's decreasing the viscosity of it, and you get a runny nose, right? And so it's kind of interesting to think about, oh, that's all because there's a canal or a drainage pipe that runs from my eyes into my nasal cavity. A couple of the other exterior or external or superficial structures we're going to talk about are the sclera. So the sclera is going to be the white portion of your eye, and it's going to be mainly the attachment point for muscles. The cornea and the lens are two of the major optical portions of your structures of the eye, and they're in charge of focusing light. The iris and the pupil are both involved with focusing light or maintaining an appropriate amount of light to the eye. And so the iris is going to be that color portion of your eye, and it's going to be connective tissue and smooth muscle. And really what that's doing is controlling the size of the pupil. Right? And so if you, iris, since it's the color portion, you can look, if you have brown eyes or blue eyes, the actual whole range of eye color starts with, if you're albino and you have no pigmentation there, it's going to appear pink. But it goes pink all the way to blue, ending in brown eye, and really every other color besides brown is going to be a mutation of some sort. If you look in the animal kingdom, there's very few species out there that have eye colors that are varied from brown or from a dark color. Humans, huskies are another one that you actually see that blue eye mutation in pop up a lot. But it's kind of interesting to think about, well, where does that mutation come up? Why is it important and things like that? And just again on the cornea, that's whenever you hear about LASIK surgery, you're actually changing the structure, anything inside the eye. You're not affecting the lens. You're actually just reshaping the cornea so that it reflects and processes light cleaner or more effectively. So there are 6 extrinsic eye muscles that attach to the sclera. And you have 3 cranial nerves that are going to help coordinate those movements. You have the ocular motor, the trochlear, and the abducens. And each one of those is in charge of 1 or 2 or 3 muscles that really focuses or allows the eye to move in a wide range. It's pretty impressive how many angles you can move your eyeball. And it's all because they're being pulled in different ways. And so there's a little schematic in the bottom left corner there that shows which muscles and resultingly which cranial nerves are associated with certain eye movements. The conjunctiva is going to be this transparent mucous membrane that covers the eye and actually covers the sclera and comes in, is actually a part of the cornea in a sense, and it rolls into and underneath the eyelids, and depending on where that is or where specifically in the eye that is, it can be made from either non-keratinized simple columnar or non-keratinized simple squamous. And if you remember that your eyes most of the time are pretty moist, right? Just like your tongue. Your tongue's another example where you see non-keratinized. In that case, it's stratified squamous, but it's the same basic principle. Your tongue needs to maintain a kind of moist layer to it. And so the conjunctiva is this layer of protection. It has all these goblet cells that help secrete mucus to, again, offer some layers of protection. And if you think about pink eye or conjunctivitis, right, that's an infection of the conjunctiva. It's going to affect some of those mucous membranes or those mucus, those goblet cells that hold mucus. So that's why you get that eye kind of gets all red and mucousy. It's because you're affecting or you have an inflammation in this specific structure. So moving into the optical components within the eye, we already mentioned that the cornea is the primary structure and the lens also is dealing with fine-tuning. The aqueous humor and the vitreous humor are 2 kind of fluids that exist, and really the only difference between them is their location. So the aqueous humor is located in the what's called the anterior chamber, which is going to be a bit in front of the pupil and the iris, and the vitreous humor is going to be located behind it. And humor, what that really means is talking about body juices, right? Humor is just fluid. So if you heard the term good sense of humor, what that means is all your juices in your body are in appropriate amounts. So everything is good sense, and so you're happy and you're healthy and all is good. That's why if you are they used to believe that if your humors were out of whack, they would put leeches on you or do other things to drain some of the bad humor from you and to get those proportions of all the body juices in the right amount. So here's a kind of a schematic walking your way through a little bit more detail the anterior portion of the eye. Right, so you can see the anterior chamber is filled with aqueous humor. The posterior chamber, or not really the posterior chamber, but the behind the lens is the vitreous humor. Those things it doesn't really matter how you spell humor. Sometimes it's spelled H-U-M-O-R. That's the American version. The European or English version has the extra U in there. Either one is acceptable. The vitreous humor is really there for kind of protective functions. The aqueous humor does that same thing. It's really about helping to keep the eye healthy, helping to maintain kind of the appropriate pressure within the eye. The vitreous humor is really responsible for helping the retina stay attached to the back of the eye. Right, so these are all, they're both really involved in just maintaining a healthy eye. You can kind of work your way in. You can actually see some of the smooth muscle that's associated with the pupil, right? So remember your pupils dilate. Well, you don't really have control over that. So if you have a muscle where you don't have control over it and it's not located in your heart, then that involuntary muscle is going to be smooth. So the actual structure that produces aqueous and vitreous humor is what's known as the ciliary body. If you remember that cilia are hairs, these are kind of have this hair-like or rigid appearance, and that image on the right showing the lens and the ciliary body that's a sheep eye, and you should be able to see that kind of dark, rigid hair-like ring pretty easily. And so the ciliary body is, again, all about producing and kind of reabsorbing the vitreous and aqueous humor. So those are all just the basic structures that make up the eye. We really haven't talked about the meat yet, which is the retina, and we're going to get to that now. So what is vision, right? So vision is just going to be the perception of this electromagnetic radiation or light that's going to be reflected off of objects in the environment. And so we have a very specific range of wavelengths that we can perceive. And that's not to say, again, that we can't actually, we're not receiving those stimuli. We're still being hit with those things. We're still being hit with UV ultraviolet radiation, but we don't have the structures capable of to allow our brains to perceive it. Right? And it's the same idea that you see in the cochlea, where the cochlea, you have a certain range, 20 to 20,000 hertz. We are still receiving stimuli, we just don't have the structures capable to perceive it. So what's happening is that the light's passing through all this stuff, hitting the retina, going to the posterior portion of the retina, and then causing this photochemical reaction that allows, that triggers that nerve signal to the brain. So light enters through the eye, through the cornea, and then through the lens. It flips, and then so you're going to have a mirror image on the back of the eye, and it's going to stimulate these rods and cones. Right? And so I'm sure you've heard rods and cones before. We're going to talk a little bit more about what exactly those are, but the rods and cones in the back then trigger a series of events that then send the information to the optic nerve. So here's another schematic. You can see that yellow arrow where the light's entering. It's passing through these ganglion cells, bipolar cells, through the meat of the rods and cones to these photoreceptors, this receptive field portions of those rods and cones. It's triggering that and sending that information back to the rods and cones, back to the bipolar cells, back to the ganglion, and then out to the optic nerve. Right? And so it's kind of a weird design in the sense that light has to pass through all this stuff to trigger this. All the cells, the trigger cells, are as far away as possible in terms of where they are located within the retina, but it works. It's not that efficient of a system, but it works. The other thing that you should note is that, well, you've heard the term blind spot, but what is that? So the blind spot is just going to be the absence of photoreceptors. And where you have an absence of photoreceptors in your eye is where the optic nerve enters or leaves the eye, right? So you don't have any photoreceptors there because that area is filled up with the optic nerve. And so if you have no receptors there, you're not going to be able to perceive light. But what's really cool is that your brain doesn't really care about that blind spot, more or less, but it's going to fill in with information. So it'll take everything else that you can see and kind of fill in that one little spot in there, so you're not actually missing information. It's just making sense of are taking information that's already existing in the environment and kind of putting together a complete puzzle. So a little bit more detail with rods and cones. Cones are going to be dealing with visual acuity and color. Rods are going to be dealing mostly with motion and dim light. A way to think about this is that rods are kind of more heavily located in your peripheral vision. So if you actually look out the side of your eyes, if you look straight ahead but kind of focus on information coming out from the side, you're actually able to pick up and see more stars out of that perspective because you have more rods located there. And it makes sense for rods to be in the peripheral because they're picking up motion, right? So you imagine you waving your hand side right by your head, you can't really see what you're waving, but you can see that there's some motion happening. And so whenever you see motion, your instinct is to turn and orient towards that motion, and that's where you're going to pick up or you're going to cue in on those cones, which are really where your visual acuity is occurring. And so what that's allowing you to do is really hone in and orient towards. And that's really what we are as a species, is we want, we're really focused with what's right in front of us. We care about what's going on to the side, but our goal is to really orient towards, not necessarily do anything with the information until we cue in on it. So we've talked about some of those external features, and we've kind of working our way into the details of the retina. I want to scale back one step into structures within the retina. And so we've talked about macula before, or we will be talking about it. And macula, again, is just a really high concentration of something. Typically it's cells, it could be cilia. We see that in the cochlea, or in the vestibular system. The vestibular system has a bunch of structures with macula in the name, and that's going to be the highest concentration of something. In that case, it's cilia. Here in the retina, it's cells. And so the macula is going to be the area that's really responsible for sharp vision. So that high visual acuity is really linked in with the macula. And within that kind of focus area, you have this even higher concentration of cone cells. Remember that cones are responsible for visual acuity, also color. Within that macula, that structure is called the fovea centralis. And the fovea centralis, again, is going to be the highest concentration of cone cells. That's going to be where you're going to be able to pick up, you're going to be most sensitive to light signals. The other structure that's labeled there is the optic disc, which is going to be it's going to associate with the optic nerve. You don't really need to worry about it too much right now. And so we kind of hinted at this when we first started. Why do bunny rabbits and deer have eyes on the side of their head versus a cat or a mongoose, which have their eyes oriented more towards the front? What's the significance of that? It's kind of important to think about, well, how do the structures or something as simple as just the orientation of the eyes really kind of clue in into what type of animal that is? And there's also this wide range of pupil shapes. So why is a pupil on some animals kind of wavy or almost bar-like, and why in a cat do they run vertical or not circular? What is the significance of that? And so you should be able to think about some of the basic functions of what the pupil does and how that would translate into different behaviors or different strategies to utilize those structures. And the last thing I'd like to end with is this kind of optical illusion. Right? So this is a mask of Einstein, and it's always spinning clockwise. It doesn't look like that, right? It's weird. It looks like it's spinning counterclockwise, and then it flips over and spins clockwise. And I know what's going on here, and it doesn't matter that I know what's happening. It's your brain is still going to override your visual cues, and that's really kind of an interesting thought. you're receiving information. Your eyes aren't interpreting that information. They're just sending signals to the brain. And the brain is using all this previous experience of shadows, of seeing people, and it's just trying to make sense of what's going on in the world. And so we really don't see a lot of concave faces. We only see convex. Right? And so your brain wants to say, oh, this is a face, but it doesn't make sense, and you don't really get the context that it's the inside of a mask until that other side flips around, and then you say, oh, okay. And then you can watch this for hours, and your brain is still not going to be able to pick that up. It just doesn't make sense. And we'll talk about this a little bit more in lab, just how our brain uses shadows and laws of lines and walls, and to make sense and to base these things off of. So it's pretty wild just to think about how complex that is, right? So if you think about it, that's just all based in electrical signals, right? So it's taking light, and then that information is being stored in some way in electrical signal in the brain, in the neurons and the axons, and then that information is then trying to make sense. It's trying to fit it in all into a pattern. It's kind of cool to think about. And again, we'll talk about this a little bit more in lab.